What is an operon?
An operon is a group of genes that are transcribed at the same time. This means that RNA polymerase reads and creates mRNA strands for all of the genes simultaneously.
An operon consists of three main parts: the promoter, the operator, and the structural genes.
The promoter is the region to which RNA polymerase binds, which initiates the process of transcription.
The structural genes following the promoter region and the operator can be transcribed and then translated into enzymes.
The operator is like the on and off switch of the gene, as it is the point to which the repressor protein binds.
If the repressor protein is present, it blocks RNA polymerase from reading farther down the operon and therefore expressing the structural genes. However, the repressor can be disabled by being binded to by an inducer molecule, which opens up the pathway and allows RNA polymerase to encode the structural genes and complete transcription, the first step of protein synthesis.
The LAC Operon is inducible, meaning that its automatic setting is for the gene to be switched off due to the presence of a repressor. This is because when the repressor is produced by the regulatory gene, it is initially in active form, so it immediately binds to the operator. This means that the structural genes are not normally transcribing and translating into proteins.
The E. coli bacteria is able to use different sugars for energy when glucose levels are low. One of these sugars is lactose, a disaccharide containing glucose and galactose.
The LAC operon is responsible for encoding the enzymes needed to obtain and absorb energy from the lactose in the local environment. In order to achieve these two functions, two enzymes are needed: beta-galactosidase and lactose permease.
Beta-galactosidase hydrolyzes lactose into its two monosaccharide parts, glucose and galactose.
Lactose permease is a transport protein that is embedded in the E. coli membrane and helps to transport more lactose into the cell.
In this diagram, Z is beta-galactosidase and Y is lactose permease.
Because the LAC operon is inducible, the repressor that is bound to the operator needs to be lured away to unblock the gene and allow for the expression of beta-galactosidase and lactose permease.
So how can the repressor get lured away from the operator? In the case of the LAC operon, lactose is the inducer.
It is called an inducer because this action causes or induces the production of proteins.
When lactose binds to the repressor is switched off into its inactive state, causing it to detach from the operator.
When the repressor detaches from the operator, RNA polymerase is free to continue reading and expressing the gene, consequently transcribing beta-galactosidase and lactose permease into mRNA. Each group of 3 consecutive nucleotides is called a codon. mRNA travels out of the nucleus and into the cytoplasm, where a ribosome calls in tRNA with the corresponding anticodon and the amino acid that matches. This process is called translation. This is how the mRNA of structural genes like beta-galactosidase and lactose permease become a string of amino acids that can function as a protein.
To review what we learned so far, let's look at our analogy to better understand the process of gene regulation. Let's say that the strand of DNA containing the LAC operon is the road.
RNA polymerase, which wants to continue down the operon to express beta-galactosidase and lactose permease, is represented by a robber.
The robber wants to run down the road to make a great escape from his latest crime. However, he is stopped in his tracks by the police officer standing in the middle of the road, blocking his path. The police officer represents the repressor protein that blocks RNA polymerase from continuing to read and express the gene.
This scenario with the robber being stopped by the cop represents gene regulation in which lactose is not present, as there is no inducer to lure the repressor away.
However, if a delicious pink frosted donut is within reach, the police officer is overcome with temptation and leaves his post on the street to enjoy his afternoon snack. At this time, the robber has nothing keeping him from continuing down the road, so he keeps running.
This represents RNA polymerase continuing to read the operon and express beta-galactosidase and lactose permease. This happens when the inducer molecule, lactose, is present to lure the repressor away from the operator.